All right, good afternoon. Welcome back. Today we're going to talk about antiviral drugs. Last time we talked about how vaccines can prevent an infection, and they have very little effect once the infection has begun, with one exception. What is the one exception? Rabies that I told you about, where you can be bitten by a rabid animal and still be immunized in time, because it takes a long time for the virus to get from the bite to the brain. Now, if you're bitten on the hand, that's good, because the virus has to go up your arm and into the CNS. But if you are bitten on the face, you have less time, because that's closer to the CNS, and the virus will get there quickly. But what we can do, if you are infected and you do not have a vaccine, we can give you antivirals, which can stop an infection once it has started. Unfortunately, we don't have many antiviral drugs. People have been working to discover them for 50 years, and the collection is very small. About 100 antivirals are available on the US market. It's different elsewhere, but this is in the US. And most of them are against viruses that cause persistent infections, like HIV, hepatitis C virus, and herpes viruses. And why is that? Because you have a lot of time to diagnose the infection and to treat it, because by definition they go on for a long time. And this is a lovely graph that shows you how most of the antivirals we have were discovered in the last 20 to 25 years. HIV is in red. You can see most of our antivirals are against HIV, and then there's some against herpes viruses, influenza, and hepatitis C virus. So this is very little number of viruses compared to all the ones that we've even just talked about in this course. Here's the whole armamentarium, more or less. There are a few missing, but so this is a 2016 article, I believe. This is most of the antivirals we have against retroviruses on the top, so that would be HIV. And then DNA viruses, and these are papillomaviruses, hepatitis B virus, cytomegalovirus, herpes simplex virus, and varicella zoster virus. And you can see some of the HIV drugs overlap in use with Hep B. Why would that be? What's in common between HIV and hepatitis B virus? Reverse transcriptase. And so some of the drugs for HIV reverse transcriptase can inhibit HBV reverse transcriptase. And then on the bottom of our RNA viruses, hepatitis C virus is the one persistent infection here. And then we have two acute virus infections, influenza virus, which is tough to give drugs for because you have to work quickly, but it's so serious that we still have them, and respiratory syncytial virus, because this hits kids in their first 6 weeks of life after birth, and there's no time to vaccinate, so you have to have some kind of antiviral drug, and so we have a few for that. But what is coming down the line, and very interesting, is probably in the future we're going to immunize the pregnant mother, and the antibodies will cross the placenta and protect the infants. There's more and more evidence showing that that will actually work. So why are there so few antiviral drugs? Number of reasons. Things that interfere with virus replication often affect the host cell. These we call side effects, and they are unacceptable. And that is because, as we know, viruses are obligate intracellular parasites. They depend on host functions of various sorts. It's very difficult to target a virus-specific function without overlapping out of the host. You will see today we do have some drugs that hit what are apparently virus-specific enzymes and so forth, but often you have side effects because there's some overlap with a host cell function. And in other cases, it's very hard to make antivirals. For some, it's very difficult to propagate them in culture. For many years, hepatitis B virus and human papillomaviruses couldn't be propagated in culture, so very hard to make an antiviral because you don't have a way of assaying inhibition of infectivity. And sometimes there's no animal model. So smallpox, there's no animal model that's acceptable for smallpox. The FDA requires you to have results in 2 animal models for them to license an antiviral drug. They made an exception for smallpox, the US paid to have 2 antivirals against smallpox developed precisely for in case of bioterrorism, because this is a prime virus to be used for bioterrorism. Very few people are immune anymore, and if it were released, we need to have some antivirals to deal with it. So the FDA made an exception and didn't require 2 animals, but for most other viruses you do, and it's not always available. And sometimes viruses are very dangerous, and you can't really work on them in a drug screening facility. Ebola virus and Lassa viruses need to be worked on in BSL-4 facilities, where you have to wear a spacesuit and go through lots of procedures so you don't get infected, and you can't do drug screening that way. So you have to develop other ways to look for antivirals against those kinds of viruses, but it restricts the number that we have. A third reason is potency. A compound has to block virus replication completely. And that's different from other pharmaceuticals. You know, you take aspirin, it's probably not inhibiting its target 100%. You don't have to. You probably only need a lot less to get rid of your headache. Most standard pharmaceuticals are okay with partial inhibition of enzyme activity, but not viruses. This is not acceptable. And giving an antiviral that only partially inhibits replication is a recipe for selection of resistance. You allow the virus to reproduce in the presence of the drug, and resistant mutants will quickly emerge. And that's shown on this graph here, where we're looking at virus load versus time. We're giving drug in the beginning, and we have given a low dose, an intermediate, and an optimal dose. The optimal dose is what you want people to have, which inhibits virus reproduction. But if you give an intermediate or a low dose, you can see the virus is able to reproduce. So this is unacceptable for an antiviral. And so you have to find drugs that completely inhibit, and that is very difficult to achieve. Another reason why they're so expensive. And also another issue was illustrated way in the beginning when I showed you that most of our antivirals are against persistent virus infections. Acute viruses are hard because they come and go so quickly. By the time you feel sick, in fact, it's often too late to impact clinical disease. And so you have to give them early in infection. We just don't have diagnostics that can work that quickly. And it's actually a chicken-egg situation. Companies do not make antivirals for acute infections because there are no rapid diagnostics. But someday there will be, and we will have more antivirals. I envision a world where scanners are built into mirrors. So when you get up in the morning, you stand in front of your mirror, it's going to scan you and tell you if you have an infection. And if you do, it will email your physician, and you can pick up your prescription on the way to work. Or it will say, no viruses detected today. Well, it will say, no viruses other than the usual. But we don't have that yet, so it's very difficult to make these. And in fact, the one antiviral against an acute infection, influenza, if you don't take it within 48 hours of feeling your first symptoms, it's not really useful. Now you may say, why don't we just give people antivirals all the time? In general, we don't like to give healthy people drugs. Not a good idea for many reasons. The only exception is PrEP, pre-exposure prophylaxis, where we now realize we should give certain people at high risk for HIV infection drugs almost continuously to prevent infection. We'll talk about that later. And we don't have any broad spectrum antivirals, which would solve part of the problem, because you can imagine if it's wintertime and you go to a physician with fever, cough, or sore throat, probably influenza, they could give you a broad spectrum antiviral, but we don't have it. I think we will probably one day, but those are harder to make because every virus is slightly different. And so diagnostic issues are also hindering this. And as I said, a company is not going to make a diagnostic for an acute infection if there are no antivirals, because no one's going to be diagnosing that. Let's take a little look at the history of antiviral discovery and how it's progressed to where it is today. The first search for antivirals began in the 1950s. We had already known that we could make antibiotics, since the 20s and 30s that were effective against bacteria. And so chemists looked at derivatives of the sulfonamide antibiotics. They made chemicals that were active against pox viruses, which was still a problem after World War II. And then in the 60s and 70s, companies began what was called blind screening. And they just look randomly for chemicals with antiviral activity. And I'm going to show you what that means in a moment. In blind screening, companies take libraries of chemicals, so companies that are doing drug research of all sorts, amass a library of compounds that they've produced for different purposes, and they've been screened, and sometimes they're not useful. And they keep them because they can rescreen them for other things. And so they can use those. And often companies will have hundreds of thousands of such compounds in vials stored away. Many times companies look for natural products. They get microorganisms that grow in soil, and they look for any antiviral compounds produced. My wife used to work at Merck, and years ago, whenever we went on vacation, she'd bring a little kit with her, which consisted of a shovel and some Ziploc bags, and she would take dirt specimens wherever. And then she had this thing which would tell you the GPS coordinate of where she took the specimen. So she'd write it down in case they found something in it, they would want to go back to find it. And so you could find lots of chemicals in natural products. So what you would do is screen your libraries or natural products for the ability to block replication of viruses in culture. So you just test a whole bunch. It was blind because it wasn't a focused assay. And then you might get hits, which would be a mixture usually of things that block virus replication. You'd then do some purification, and maybe you would then get some results in animals and develop these compounds into leads. You take the hits, which come right out of the first screen. You decide what compound it is. You begin to modify it to make it more potent, to reduce toxicity, make it more soluble, make it more bioavailable, which means if you take it by mouth, it goes to where you need it to be. And other pharmacokinetic properties, you don't want to have a short half-life, you want it to last a long time and go to the right tissues and so forth. These resulted in thousands and thousands of molecules being screened, but very few antivirals being developed. One exception was this compound called Symmetrel or Amantadine, which is right here on the right, which was approved in the 60s for the treatment of influenza A virus infection. But we didn't know its mechanism of action until many years later, and I'll talk about that today. We figured that out in the 1990s. Today, almost all circulating influenza viruses are resistant to this drug because it's been abused in many places, not just in humans, but in animals. Farmers often feed it to pigs because pigs can get influenza, and when they do, they don't gain as much weight. So just like they feed antibiotics to animals, they give them this, and it drives resistance to the antiviral. Today, the drug discovery programs of companies are very different. Recombinant DNA technology and very sophisticated chemistry have revolutionized the process. We can target drug discovery very specifically. You can clone genes, you can express them in organisms, you can purify the products and do assays in vitro for enzymatic activity. And because we know the reproduction cycles of viruses, we can say, okay, we're going to target this part or that part or another part. And even if you can't grow the virus in cell culture, you can develop an assay which will allow you to screen for inhibition. And so no more blind screening. Nobody takes mixtures of chemicals and just screens against a lot of different viruses. It's always very focused. We still use chemical mixtures. Again, many companies have them, but you can now purchase libraries. You could say, sell me all the chemicals that are FDA approved, and I'm going to screen them against my virus. And that's kind of an interesting way to do it, because if you get a hit, it's quicker to get FDA approval for something that's already approved. And so we have reproduction cycles, as we've learned in this course, for all of our viruses. We know the different steps in the reproduction cycle, and we can design inhibitors. We could decide which step we're going to attack. We could look at attachment, for example, and uncoating, mRNA synthesis, protein synthesis. DNA and RNA replication is a big target because the enzymes, they are clearly related to cellular enzymes, but you can design compounds that have some specificity in even assembly. The path to getting an antiviral drug is difficult and very expensive. And it starts with identifying a medical need. As I said last time, for vaccines, you need to know that the virus is a substantial human pathogen. And once you do that, you have to figure out what you want to target. let's say you have a gene in mind that you want to design an antiviral for. You first have to do a proof of principle and show that that gene is essential. If you don't do that, you could develop a compound that inhibits it, but then in the end it wouldn't block virus replication. So that would be foolish. So very early on, if you can make a deletion or a mutation in the gene and show it's essential, then you could go to your bosses in the company and say, this is a good target. And that involves some research, of course, which could go on beforehand in an academic laboratory. Then you have identified a target. You could start doing screens with your natural products or your compound collections or other sources of inhibitors. You can design a variety of screens, cell or mechanism based, which we'll look at today, or even in silico. You could design a screen entirely on a computer. If you have the structure of a molecule, you could tell the computer, tell me what kind of small molecule would fit into the active site. So that's an in silico screen, and then you can get suggestions and start to make those and test them. All of that will give you hits, which then you bring along to lead compounds. You modify the hits to make them more potent, to make them less toxic, and so forth. And some of the things you need to address. Will it get to the right place in the body at the right concentration? That's bioavailability. Will it persist long enough to be active? Pharmacokinetics. And will the compound be safe? You test that in cell cultures. And if you put a compound in a cell culture and the cells die, that's the end of that. And then if you get things through cell cultures, you put them in animals. You ask, are they potent? Are they safe? Do they go to the right place? Someone told me once that the most successful people in big pharma companies are the ones who know to stop working on a compound. Instead of taking it as far as you can, learn very early on when a compound is not going to work and get rid of it. So it's very expensive to make an antiviral. We estimate $100 to $200 million to bring it to market. And that's because there's a lot of time and a lot of steps. We're showing 10 to 15 years here for a typical antiviral. You start with hundreds of thousands of compounds, and at each step in the development, you have to throw more and more away. You have to see if there's an antiviral effect in cells, and some of them won't have any. So you throw a bunch out, and then toxicity, you throw a bunch of those out, antiviral effect in animals, toxicity in animals, and then here this is a little misleading. It looks like we're putting 10 compounds into humans, but that rarely happens. Usually you end up with one candidate that goes into a human clinical trial because they're just too expensive to do multiple ones, at least at the same time. And if you can get through your clinical trials, it can be approved for use, but it's very expensive. And so when you wonder about the cost of drugs, antiviral drugs in particular, I can defend because the work to get them is very costly. So here's a nice timeline from discovery to the clinic. You have 5 years of discovery and synthesis. Preclinical means work in cell culture and in animals, and the results you need to file an application to the FDA. So you need 2 animal models, you need cell culture results, and then you file what's called an IND, an investigational new drug application to the FDA. You say, I have this drug. I was recently at a biotech company, and they had just filed an IND for an antiviral, and the documentation took up 3 shelves of notebooks, thousands and thousands of pages. And then if it's approved by the FDA, you can then go into humans. You would have a phase 1 trial where you have a small number of people. You just test the safety of the drug. You don't really looking at efficacy, but you want to make sure it doesn't cause any side effects. And the people that do that are rather brave, I think. Phase 2 and 3 are usually dosing trials and efficacy. How much do we need to inhibit whatever it is that we're looking at? And if this is a virus infection, you have to make sure, typically for the phase 3, that these are people in an area where they're going to get infected. And that could be influenza or HIV or any virus for which you're making the antiviral. For HIV in particular, it's quite interesting. You have to find people who are at risk, and then you have to tell them not to engage in risky behavior, i.e. have unprotected sex. However, if they don't, your trial will fail. So you're depending on people not listening to you. But you are bound to tell them the risks ahead of time. You shouldn't do this, and that way it is ethical. And then, after these 3 trials, the FDA reviews the drugs, results in people, and may approve it or not. But of course, even if it's licensed and sold to the public, you have ongoing safety reviews because now it goes from a few hundred people into maybe millions of people. And there you can see things you didn't pick up in the clinical trial. So you always have to have a constant evaluation of what's going on. This can take a long time, as you can see, up to 20 years. So let's look at a couple of screens that you can do. I'm going to just show you 2 or 3 to give you an idea. Here's a mechanism-based screen. Let's say you have a protease, a viral protease that you know the substrate for, and you want to make an inhibitor. And so here is the substrate, ABCD, which you can synthesize, and you couple it to a bead on one end and to a fluorescent source, a fluorescent compound, something that's going to emit light on the other. And if the protease, if you've incorporated the cleavage site, the protease will separate the bead part from the other part, and so you can centrifuge the bead part out and then look at the supernatant for how much fluorescence, say, there is here. And here's an example of that assay, fluorescence intensity of the soluble peptide with time. So the protease is cleaving it, and as you centrifuge out the beads, the fluorescent part remains in the soup. And now if you have an inhibitor, you can simply assay the effect of that inhibitor of the protease on the release of fluorescent substrate into the supernatant. Very simple assay that can be automated and done with lots and lots of chemicals. We call that high throughput. You could screen tens of thousands of chemicals in a day. So that's a mechanism-based screen because we're looking at the protease cleaving it. But we can also do this in cells. And here's an example of an assay set up in bacteria. We have bacteria which are made tetracycline resistant because they carry and produce this tetracycline efflux pump. It's a multi-pass transmembrane protein. And if you treat bacteria with tetracycline, which would normally inhibit their growth, this pumps it out so the bacteria remain resistant. And you have engineered this pump so that it has an HIV protease site in one of these loops in the inside part of the cell. And so the HIV protease, when produced in the bacteria, will cleave that loop, and it will render the efflux pump inactive. And so these bacteria will be inhibited by tetracycline. And now if you add protease inhibitors, you will block the protease. Some of them may block the protease, and the readout is growth of colonies on agar. So the bacteria are now resistant because the HIV protease is not cleaving the loop, which is in the tetracycline efflux pump. So you can look at colonies on a plate as a readout of the efficacy of an antiviral. And so this is just 2 of the many, many ways that people design screens. The key is you're looking at a specific part of the cycle, here the protease in these last 2 assays, and you can automate it so that you don't have to do it by hand. And so you can do lots and lots of compounds in a day, 10,000 or more, so that's high throughput. And this can be applied to chemical libraries. It can be applied to natural products. You can do combinatorial chemistry. The illustration on the right shows how you can combine linkers and fragments chemically in these matrices to make lots of different chemicals, all of which are slightly different. And you can test thousands of these different arrangements of these subunits. You can do structure-based design. You can have the 3-dimensional structure of an enzyme, a protease, say, or a polymerase, and you can see the active site and try and design things on the computer that might fit into it, and then make those chemicals in the laboratory and see if they work. And as I said, you can do in silico screening, where you would take the model and the computer would run thousands and thousands of iterations of the compounds through it and see which ones fit best, say, as a measure of which ones would be potent. So today, the antiviral lab is roboticized. In fact, when I went to this little biotech company a few weeks ago, I walked into a room and there was a robot like one of these. It is essentially an arm that picks up the plates and moves them around. It moves them into racks. It can pipette compound into each well. It can set up the assay into each well. So here are the wells being filled by the robot, and you can use plates with lots of wells. You can up to 1,536 wells. The robot will pick it up, we'll fill it up, and then we'll place it in an incubator. We'll do any kind of manipulations that need to be done. So you could set this up on Friday afternoon and go home and watch the results come online over the weekend. And that's what a lot of people do. So it's all roboticized. You can go through lots and lots of compounds, and you get ones that look like they're inhibiting, and you proceed with those further. Our first question is, we have many antibiotics but fewer antivirals. What is a reason? Robotic screening is slow. There are few serious viral infections. Resistance is a problem. Antivirals must be potent, or all of the above. The answer is antivirals must be potent, which most of you got. Robotic screening is not slow. It's very fast. You can do tens of thousands of compounds a day. There are few serious viral infections isn't correct. And resistance is a problem, but it doesn't prevent you from making antivirals. It prevents you from continuing to use them afterwards. So the potency is the real issue here. And that's why, of all those choices, of course there are other reasons, as I've told you, but they're not on the slide. Let's talk about resistance. You can make any antiviral drug, and I'll guarantee that you will get resistance to it at some point. And that's simply because viruses mutate so quickly, as we'll see in a moment. They replicate efficiency, and they have pretty good mutation efficiencies. Just think of the progeny that viruses make alone. And that's, billions and billions of particles from infected cells. That guarantees enough diversity to get resistance. And this is a problem in chronic infections, where you treat people for years. You are more likely to get resistance, and we've seen that with all of these viruses. We have resistance to every drug that we have. All those drugs I showed you on the slide, there's resistance to every one of them. And that's unfortunate because we don't have that many. It's not like you can switch. Although with HIV you can now, as you'll see, because we have so many. But for many of the other viruses, particular herpes viruses, we only have a few. And if you have resistance to those, there's not much we can do. And if you're an AIDS patient that's immunosuppressed, it's probably going to kill you. And that's usually what happens, is you're killed by opportunistic, infections. And as I told you in the first lecture, there's already resistance out there to every antiviral we'll ever make in all of humanity time, because that's how many viruses there are out there. It's kind of unfortunate, but that's the reality. Yet we can get around it. Of course, when you get resistance, you can't keep treating with the same drugs. And if there's nothing else available, as I said, you can't stop the infection. We have learned that we can analyze resistance and learn how to design new compounds that get around it, and how to treat patients. For HIV now, if you're diagnosed with AIDS, they will sequence the viral genome in all the regions known to confer resistance to the compounds we have. And they will tailor your therapy to that, based on that information. There's no point in giving you an antiviral to which you are resistant, and that happens. So we'll talk about that later, but you can give people the drug that suits their particular virus. How do you get resistance? It is because viruses have error-prone polymerases, and RNA viruses and DNA viruses both have error-prone polymerases. The RNA viruses are a problem because there's no correction mechanism to repair errors that are introduced. Okay, so again, remember, both RNA and DNA polymerases make errors. As we'll see, the DNA polymerases can correct them, but the RNA polymerases cannot. So RNA polymerases make, on average, one mistake in 10,000 to 100,000 bases polymerize, and that's about 1,000,000-fold greater than host rGNA genomes in us. We also make mistakes, but they're a lower frequency because of correction mechanism. So what this means is if you have an RNA genome of 10,000 bases, you get one mutation in 1 to 10 genome, depending on whether it's 10 to the 4, 10 to the 5. You could have every reproduction cycle one mutation per genome, which is a lot. And that's why we get resistance, because these are random mutations that appear in the nucleic acid. And at some frequency, one is going to end up conferring resistance to your antiviral drug. In DNA viruses, as I said, the polymerases can repair the mistake, and so they evolve more slowly because they have less diversity. They don't have zero diversity, but they evolve more slowly than RNA viruses. So here we have a DNA polymerase copying a template. The template's in dark blue on the top, and the bottom, the light blue strand, is the product. We have 3' hydroxyl. And let's say a mistake is made, which happens, the wrong NTP goes into the active site, and you have a mismatch, there are exonucleases which will detect that and cut it out, and then the DNA polymerase will repair it. And so these are the repair pathways for DNA viruses. They do not exist in RNA viruses. RNA viruses do not have error correction pathways for the most part. There are always exceptions. The very big RNA viruses, the coronaviruses with genomes over 30,000 bases, seem to have proteins that help them maintain the error frequency at a manageable level. And they can't have genomes that big without it, but most of the other viruses don't have that. So let's take a look at some antiviral drugs and how they work and how you get resistance. And the first one is a relatively old compound to treat herpes simplex virus infections. It's called Acyclovir. It's a very good drug, and this is a nucleoside analog. What do I mean by that? And it's also a prodrug, as you'll see what that means in a moment. So here on the left, we have the 4 nucleosides. That means the base and the sugar without any phosphates. That's what a nucleoside is, right? When you put a phosphate on, 1 or 2 or 3, then it becomes a nucleotide. And we have nucleoside and nucleotide analogs, which are antiviral drugs. And for each of these 4 bases, people have made antiviral compounds that are derivatives of them. So you can see adenosine. We have 2 drugs here, which are derivatives. Acyclovir is a derivative of guanosine. You can see it possesses the base and a little bit of the ribose, but not all of it. And these compounds typically cause chain termination. They get incorporated into the growing chain, but in many cases they don't have a 3' hydroxyl. And so you can see dideoxycytidine. There's no 3' hydroxyl. So the polymerase would put this in as a C and then can't add the next base because there's no 3' hydroxyl. Sometimes there's a different chemical structure which can't be added to. Sometimes there is actually a 3' hydroxyl, but changes elsewhere in the molecule prevent chain elongation. So acyclovir is a derivative of guanosine. And there's another derivative which you'll hear about called ganciclovir. It's a 2nd generation, slightly modified, which has different applications and different potencies. So this is a prodrug, which means when you put the ointment on your cold sore with acyclovir in it, or if you take the pill, that compound that you're putting in you or on you is not active yet. It has to be activated. That's why we call it a prodrug. So here's acyclovir. It has to be phosphorylated before the polymerase will use it. And in general, nucleotides are not taken up into cells. The phosphate, whether it's 1, 2, or 3, prevents uptake through the plasma membrane. And so you have to give people nucleosides or nucleoside analogs because they will pass through the membrane. So acyclovir will get through the membrane of a cell, and then it needs to be phosphorylated. The first phosphorylation must be done by a viral kinase called TK, thymidine kinase. So that's what we have here, HSV1 TK. So that protein is only present in infected cells. It's very clever. So this drug will get into uninfected cells, but nothing will happen because it won't be phosphorylated, because those cells are not infected. They do not have HSV1 TK in them. We have many other nucleoside analog antivirals. They all have a little side effect because they're not specific to infected cells. This is beautifully specific for virus-infected cells because it has to be phosphorylated. So the herpes TK protein, again, a thymidine kinase, puts 1 phosphate on the acyclovir, and then 2 cellular protein kinases do the rest. They put 2 and then 3 on, and then it's triphosphorylated, acyclovir triphosphate, and it gets incorporated into the DNA, and it can't be elongated because it doesn't have a 3' hydroxyl like guanosine would have. And so that's why it's inhibitory. It stops chain elongation, it stops DNA synthesis, and of course it stops DNA replication. So it is a prodrug because it has to be activated by phosphorylation, and it is a nucleoside analog. This was subsequently modified to make it more orally bioavailable. So the original Acyclovir was designed for topical application on cold sores caused by herpes simplex viruses. For people with more serious disseminated infections, you have to give it to them orally. And so valcyclovir was derived. It's a ester derivative of acyclovir. Basically, what they did was take acyclovir. They did lots of chemistry on it to find this. It wasn't like they said, oh, let's do this and it'll work. But what they found is this compound, by attaching valine to the oxygen here, this compound is better taken up after oral administration. And then when it gets into cells, the valine is cleaved off. So in the cell, you now have acyclovir again. Just putting that valine on, which again was found by random chemical modification of the acyclovir, was found to make it more orally bioavailable. In other words, you take it, it gets into the bloodstream. And the acyclovir wasn't so good at that, and so this really helps it. So you may encounter valacyclovir, in particular if you have someone with herpes encephalitis, they may be given this either orally or intravenously. Unfortunately, we do get Acyclovir-resistant viruses. This is not so common, but it does happen. The resistance, as I said, arises spontaneously because of the error-prone polymerase. And there are two kinds of mutants. There are some that don't phosphorylate the prodrug, so those mutations are in the gene encoding thymidine kinase. The TK now doesn't recognize acyclovir because the TK has an amino acid change in it that prevents that, so you don't get phosphorylation. And a second class of mutants have normal TK, but the DNA polymerase doesn't recognize the acyclovir triphosphate and doesn't incorporate it into the chain. So these are resistance mechanisms. You get TK mutations or DNA polymerase mutations. And these mutations to resistance can be a real problem. They typically arise in people who are getting long-term treatment. So immunosuppressed patients who have herpes virus infections, they're given long-term treatment, you select for mutants. There are a few other drugs that you can use, but not a lot. We don't have a lot of drugs, and if you end up not being able to treat, the patients will die from disseminated herpes infection. Here's a drug for an RNA virus. This was one of the first antivirals discovered by the blind screening procedures. Amantadine, this interesting molecule here. And the way this works, it interacts with the viral M2 protein, which is an ion channel. If we go back to the structure of influenza virus, we have it here in the upper left. The HA is the blue spike, the Neuraminidase, NA is the red, and then the little purple channel there is the M2. There aren't many per particle, about 4 or so per particle, but these are channels. They let protons enter the virus particle as the virus is taken up into the cell by endocytosis. Remember, the pH of the endosome drops as protons are pumped into the endosome. Those protons flow through the ion channel. It's just a channel, it's not a pump. They get into the interior of the particle, and that loosens up the RNP so it can come out when the membranes of the virus and the endosome fuse. Now, Amantadine blocks this. It blocks the channel so the protons can't go through it, and the virus cannot release its nucleic acid, and that blocks infection. It's really a really amazing mechanism, which is why we understand a good part of this pathway, because people started using it, and they say, oh, you need this channel for uncoating, and you need protons to go through. So even though the drug isn't very useful clinically, it has told us a lot about virus entry. Here's a picture of the drug and the channel. So there's the influenza virus membrane. We're now looking at one channel formed by this tetramer of the M2 protein. It's a very short protein. 4 alpha helices make up the channel through which protons go, as you can see there. And then red is the amantadines, which bind both on the outside and the inside. They bind to specific amino acids on the channel, and they block it, so the protons can't get through. It's very easy for the M2 to undergo mutation, the gene, to change these amino acids to which amantadine binds. So now amantadine doesn't bind any longer, and the protons can go through. So that's how resistance arises to amantadine. If you are given an antiviral today for influenza, it may be a neuraminidase inhibitor. These were developed not too many years ago. If you remember the way influenza virus matures, the virus particles bud from the cell surface and then they are released. And here's a particle with hemagglutinin and neuraminidase on its surface. Remember that the receptor for influenza virus is sialic acid. So as these particles are released from the cell, they could simply be stuck to the cell surface because the cell is coated with sialic acid. That's where the neuraminidase comes into play. As these particles are budding from the surface, the neuraminidase is an enzyme that is cleaving sialic acid from the cell surface, so that when the bud forms, the particle forms, there's no more sialic acid for it to bind to, and it's released. So this, as a target for an antiviral, was shown way back in 1976 by Peter Palese at Mount Sinai. And what he did was develop a chemical inhibitor of neuraminidase, and he added it to cells infected with influenza virus. And here's an electron micrograph of such an experiment. We have the cell at the bottom here, and these little spheres are influenza viruses. And you can see they're stuck to the cell and they're stuck to each other because sialic acid is still there. The neuraminidase has been inhibited, and it can't remove sialic acid from both the cell surface and the virus particle. On the right here is the neuraminidase from the top showing sialic acid bound in it. This is the active site of the enzyme. And this information, this three-dimensional structure, was used to design compounds that would look like sialic acid. It would fit in the neuraminidase active site and inhibit it because they would not leave. And so now we have three drugs actually today that are neuraminidase inhibitors. You may recognize the names Relenza or Tamiflu. Tamiflu is the more common. It's an oral product taken orally. Relenza is inhaled, so not everyone can use that for a variety of reasons. But these are designed to mimic sialic acid. And the idea was, if we can make a drug that looks almost exactly like sialic acid, maybe we won't get resistance, because if you get resistance to the drug, you're also going to get resistance to sialic acid and the virus can't replicate, right? That's the idea. Well, of course, it doesn't actually work, but it's pretty good. It's given fewer resistances than we would have predicted. And so here's the way the inhibitors work and how resistance works. So here is a virus particle on the bottom left, and the purple Y is a neuraminidase. It's binding sialic acid on the host cell. It's going to cleave it off the glycoprotein so that the virus particles can leave. Tamiflu is this yellow inhibitor which fits into the active site, but it's not quite the same as sialic acid, shown by the differences in the structure. So sialic acid is the triangle, and Tamiflu is different. And you can get resistance to Tamiflu. We see that it's pretty low, but you can get resistance. On the other hand, Relenza, it looks very much like sialic acid, as shown by the cartoons, much harder to get resistance. Amino acid changes in the active site of the neuraminidase, shown here, lead to resistance to Tamiflu. As I said, much more rare to Relenza. And the CDC keeps track of resistance among circulating influenza viruses. It's part of their huge surveillance program. And this is this year's results. These I just downloaded the other day, since October, more or less beginning of the flu season. And, we do isolate lots of influenza viruses from people. They've tested 1,800 viruses for resistance to these two drugs. But you can see just a couple of viruses resistant to Tamiflu there, mainly the H1N1 viruses, exclusively the H1N1 viruses. And if we move over to Relenza, the one that you inhale, you can see no resistance this year. Now, fewer people are given this drug, so that may in part account for it. It's not unknown to have resistance, but we think the frequency is much lower because of the way the drug is designed. And they used to report sensitivity to adamantanes, but they're all resistant now, so they don't even bother to recommend it anymore. A new influenza antiviral was just approved by the FDA last October. So this is the first time I'm showing this, and it's called Baloxavir. By the way, there is a science to naming these drugs. There are articles online where you can see why they name them certain things. but this is approved for treatment of acute uncomplicated influenza in people 12 years of age and older who have been symptomatic for no more than 48 hours. So why such restriction? Well, 48 hours because beyond that it's useless, so don't bother to give it to people. Why acute uncomplicated influenza? Because that's what they tested it in. They didn't test it in people with pneumonia or other complications, only in people with regular influenza. So that's the only population it can be recommended for. Now, if you're a physician, you can do off-label prescription. You could say, I have a seriously ill patient with flu, and it's only recommended for uncomplicated flu, but I'm going to take the risk of giving it to this person. So that happens quite often. This is a really interesting drug. There it is. Dihydroxy chicken wire is the name. That's actually a colleague of mine. He looks at compounds like this, and that's what he calls them, because this looks like chicken wire. But this should be difluoro chicken wire. Right? There's no hydroxys there. What it does is it inhibits the endonuclease. If you remember, the influenza polymerase, when it makes mRNA, the primer is derived from host cell mRNAs. The viral endonuclease cleaves host cell mRNAs to give roughly 13 base capped primers, which then prime mRNA synthesis. So that reaction, that cleavage is done by a viral endonuclease. It's in the virus particle. And so they screen for an inhibitor of it, and that's what this is. And this works pretty much as well as the neuraminidase inhibitors. Are we going to get resistance to this? Of course. The answer is always yes. And there are going to be mutations somewhere in the endonuclease, probably at the active site, but perhaps elsewhere as well. And we'll find out next season when the CDC starts to screen for those. Next question. Resistance to which antiviral would involve amino acid changes in a viral enzyme? Acyclovir, Amantadine, penicillin, or all of the above? The answer is acyclovir, which most of you got. You get resistance by changes in the TK, thymidine kinase, the kinase which is an enzyme, or the viral DNA polymerase. The amantadine resistance, that is a channel. It's not an enzyme. And penicillin, of course, is not an antiviral, so that was just thrown in there to confuse you, and it's not all of the above. There are some antivirals that have been developed for picornavirus infections, like rhinoviruses and polioviruses and other enteroviruses, but we don't use them because you get resistance very rapidly, and we can't really diagnose those infections quickly. But they've been useful in the laboratory for a variety of reasons, so I want to tell you about them briefly. One set is called the Win compounds. These are developed at a company called Sterling Winthrop. And what they do is they bind in a pocket which is in the capsid of the virus. So here is part of the virus particle here. There's the five-fold axis of symmetry. And there's the receptor fitting in to the groove on the particle surface. And so this little pocket is on the floor of what we call the canyon. And it's blown up here on the lower left. Now, normally, this pocket is occupied by a cellular lipid. It's shown on the right here. So there is, in that pocket is shown a cellular lipid bound. So again, on the upper diagram, that's the pocket. Here it is on the actual proteins below. The viruses, as we grow them up, have lipid in them. And when the receptor sits down on these viruses, it pushes the lipid out. And that gives an open space so the particle has room to undergo conformational change so it can release the RNA. What these drugs do, and here's one drug on the lower left here, they displace the lipid and fit into the pocket, and they fit so tightly they don't leave. So when drug-bound viruses bind receptor, the drug remains inside, and the virus can't uncoat, because it needs that open space to be flexible enough to uncoat. So these are uncoating inhibitors. As I said, it's very easy to get mutations in the amino acids lining the pocket that prevent the drug from binding, so they're clinically useless. But they have explained to us how uncoating works. So a study of this drugs bound to viruses let us know that flexibility was needed to release the RNA. I want to show you how these were initially discovered way back in 1985. Here's the original drug on the upper left, which was picked up in a screen of libraries. They were screening for inhibitors of polio-like viruses, and they picked this inhibitor up. And what we see here in the middle are attempts to modify the compound to make it more potent. You can see various modifications. There are lots of molecules here which are derivatives of the original. They're flipped around, as you can see. And then for each one, they tested the minimal inhibitory concentration, which is in micrograms per mil. And you can see this one at the top seems to be the original compound that has an MIC of 12. And they made a change to it, and it's inactive. Made a different change. It's better, inactive, inactive, the same, inactive. So you can see these are just random changes that the chemists make, and they test them for activity. And if you get better activity, they will then move on with those. Another way of doing this is to change the number of carbons between the phenol ring and the rest of the molecule. So you can see here N it can be 3, 4, 5, 6, 7, up to 10 carbons in between, and they test the MIC. And you can see with 3 it's inactive, with 4 it's 25, but then if you go to 6, it goes back down to 6. You put more in, it's inactive. So there's actually no logic to any of this, but you just pick the one with the best activity, and you pick the one with the best least toxicity and other properties that you like. And that's the way these compounds are modified. So the chemists have a big role to play in companies looking for these, not just antivirals, of course, but any kinds of pharmaceuticals. And so if you like chemistry, this is what you can do. There have been more recently, drugs developed against hepatitis C virus, which we talked about as a persistent virus infection. Originally, patients who were hep C positive were treated with a combination of ribavirin, which induces mutations in the viral genome, and interferon, combination of both. But of course, the interferon makes patients very sick. It gives you flu-like symptoms all the time because you're taking the interferon all the time. So people would stop taking it, and they would have viral rebound and so forth. So we needed to develop other inhibitors, and that was done by studying the viral reproduction cycle and learning what the different proteins do. So we found, for example, that there is a viral protease, NS3, which is responsible for cleavages of many of the bonds in the polyprotein that's produced by translation. So people produced this protein, they determined its three-dimensional structure, they saw where the active site was, and then they devised inhibitors by some of the screens like we've talked about today, and one of them is called telaprevir, and this has been used now to treat hepatitis C virus. They also have made inhibitors against other proteins. NS5B is the RNA polymerase of the virus, so they made inhibitors to that as well. So, hep C was discovered relatively recently, and we needed to do a lot of fundamental work on understanding how it replicated before we could make these inhibitors. Here's a list of some of the inhibitors available now for hep C treatment. You can see the target. The polymerase is a target of several drugs. They can have either nucleoside or non-nucleoside inhibitors. They're inhibitors of another protein, NS5A. They're inhibitors of the protease. And more importantly, at the bottom are combinations of these drugs, which is now how you treat hep C, combinations of 2 or 3 compounds. And because there is no latent reservoir of hep C, this is a curable infection. You can give people a regimen of treatment, and assuming they don't develop resistance, which is unlikely with 3 drugs at the same time, you will cure them of their hepatitis C. That's not the case, of course, with HIV, where there's a latent reservoir of cells with proviruses that are not replicating and which antivirals cannot touch. So cure is possible for hep C. Can we make a broad spectrum antiviral? People would like to do that. Here is one compound developed which is somewhat broad spectrum. It's called LJ001, and here's a table of different viruses of different families and different genome types, and whether the drug is active against them, shown by the pluses. What you can see is that this drug inhibits viruses that have an envelope. Whether it's DNA or RNA doesn't matter. Having an envelope is the target. And non-envelope viruses like picornaviruses and reoviruses are not inhibited. So what's going on here? Here's a picture to show you what's happening. There's the drug at the top, and then here is an assay with vesicular stomatitis virus, which is the bullet-shaped envelope particle. So here is treatment with DMSO, which is just a diluent, so it's a control. You get The particles look the same. In the middle is a compound that is not active in the same series, but you can see the particles. And then LJ001, the particles are stripped of their membrane. You're left with just nucleocapsids. So this drug trashes membranes. Interestingly, it has no effect on cells because cell membranes are always turning over. So this drug can insert into it and the cell will repair it. There are some others that have been developed. Here's one, Favipiravir. This is in development now, which is a broad spectrum inhibitor of RNA viruses. And the target is the polymerase. And you can see a lot of plus-stranded RNA viruses are inhibited as well as minus-strand RNA viruses. And so again, as I said, this is in development. I don't know if this will get approved, but it can inhibit a variety of RNA viruses. The way it works, the drug moves into the cell. It can permeate through the plasma membrane of the cell. It's phosphorylated to the triphosphate by a cell enzyme, HGPRT. And you now have Favipiravir with triphosphates. And then that can get incorporated by the RNA polymerase into viral RNA and cause chain termination or lethal mutagenesis, where it's incorporated, the chain continues, but it templates as the wrong base, so you cause mutagenesis. So it has a variety of mechanisms. And whether this gets to the market, we'll see, but it could be useful in cases where you don't know exactly what the infection is. And people are doing similar things for DNA viruses. There are broad spectrum DNA virus inhibitors. And I think it's probably not likely that we would have an inhibitor for both DNA and RNA viruses, but having one or the other might be useful. So let's now talk about HIV antivirals. This constitutes the majority of the antiviral drugs that we have. And we have lots of targets for developing drugs for HIV. We have attachment and entry. We have attachment inhibitors and fusion inhibitors, reverse transcriptase inhibitors, integrase inhibitors, protease inhibitors as well. So all different steps of the cycle. And the reason we were able to do this so quickly, the virus is only isolated in the 80s, is because people had been studying other retroviruses for many years, like Rouse sarcoma virus. And we knew all the viral proteins and how they worked and what they did, and it was easy to transpose it to HIV. The issue here, of course, is that you're infected for life. Virus is replicating continuously, and unless you are faithfully taking your antiretrovirals, you will get resistance. And so this has come up over and over again, as you will see. First drug made against HIV, Azido-deoxythymidine, or AZT. This was discovered looking for anti-tumor cell compounds. This was found to inhibit HIV. And the way it works, it is a chain terminator. It's phosphorylated. It enters the cell. Here's AZT on the left. It's got 3 nitrogen down there instead of the hydroxyl, so you could predict it's going to be a chain terminator. It's brought into the cell. It's phosphorylated by cellular kinases, not viral kinases. So this drug gets into uninfected cells and infected cells and is phosphorylated in both and will inhibit cellular polymerases as well. So it's a chain terminator. It's somewhat selective for virus polymerases. It's not a great inhibitor for cellular, but it does inhibit, and that's why we have side effects with AZT. So that was the first drug to be approved for treatment of AIDS. It's the topic of this movie, Dallas Buyers Club. If you saw that, that's the story of AZT and how initially when it was released, there wasn't enough to go around. So patients started splitting up their pills and sharing them. And that actually showed the FDA that their dose was too high. You could actually cut it substantially and still prevent infection. So the patients sharing their drugs taught them that. Unfortunately, this has a lot of side effects. Unlike Acyclovir, because it's phosphorylated by cellular kinases. It's taken orally, it's absorbed very well, but its half-life is 1 hour. This is horrible for a drug, but there was nothing else, so people were taking it. And you have to dose yourselves 2 to 3 times a day, and that, together with the side effects, was not very nice. And all of this led to the very quick emergence of resistant mutants, because the short half-life, coupled with people not taking the drug because they had side effects, allowed viruses to replicate in the presence of the drug, and we selected for resistant mutants. So very quickly, this drug became useless. It's not used anymore. These rose almost immediately after licensure. Single amino acid changes at one of 4 places in the reverse transcriptase, and these altered proteins do not bind the drug. That's the mechanism of resistance. So then people started making new nucleoside analogs derived from other bases like DDI, DDC, D4T, 3TC, etc. And people started combining different inhibitors, the use of 2 antiviral drugs, but even with 2, they still got resistance in less than 1 year. The next set of drugs developed were what we call non-nucleoside RT inhibitors, NNRTIs. So here's a reverse transcriptase. There's the polymerase active site with the little green molecules there. And just to the right of it is where these NNRTIs bind. So they bind away from the active site, and somehow they deform the molecule so it doesn't polymerize. Whereas the nucleoside analogs, they go into the active site, of course, and they're incorporated into the chain. And here are a couple of these NNRTIs down at the bottom here. Some of these are still used. Dihydroxy chicken wire. But we got resistance to these as well. They were selected relatively rapidly. Amino acid changes in any of 7 residues that are lining the binding sites for the drug on the enzyme. You don't have to make many amino acid changes. The drug doesn't bind and it doesn't inhibit any longer. So we don't use these by themselves anymore. In fact, we don't use any drug by itself anymore. Everything is used in combination therapy because whatever you throw at the virus as monotherapy, resistance emerges very rapidly. Next target was the protease, absolutely required for the production of infectious virus particles. Protease is incorporated into the growing particle. It's that yellow protein there, PR for protease. It goes into the virus particle, and then after the particle buds, the protease cleaves viral proteins and causes maturation of the final viral particle. And so if you remove the protease from the viral genome, the virus doesn't replicate. So that said, it was a good target. People learned the sequence that was cleaved by the protease. So here's the amino acid sequence of the viral protein, and that the cleavage site was identified between this tyrosine and proline. And so what was done was to make peptidomimetics, drugs that mimic these amino acids right next to the cleavage site. And one of them is shown here, Saquinavir and Darunavir. And these will bind the active site, and in fact, structural analysis showed that to be the case. There's the protease with the substrate in it. These bind to the active site. They're not cleaved because the bond is not cleavable, and they inhibit the protease. So it was a very clever application of the structure. The next series of compounds, integrase inhibitors. Remember, the integrase is the enzyme that takes retroviral DNA and cellular target DNA and joins them together. It integrates the viral DNA into host cell DNA. There's the integrase structure with double-stranded DNA. And the active site where the reactions occur are where these drugs bind. There are 3 of them shown here. They bind in the active site. They prevent integration. Again, you can't use these by themselves. You get resistance right away. And one more, because this is a really different target. This is an inhibitor of binding. This is a drug called Maraviroc, which is an inhibitor of CCR5. So we know that CCR5 on the host cell is essential for infection. We have people who lack it, and they're not infected. And so this drug was selected to bind CCR5. There's the drug on the left. Here it is shown bound to the CCR5 protein. It's on the top view, and there's a side view of the drug bound to the co-receptor. And the way this works, of course, is that GP120 of HIV initially binds to CD4, and then it binds to CCR5, which results in high affinity interaction between the virus and the cell, and it leads to cell entry. And what this drug does when bound to CCR5, it reduces the affinity of the binding of the virus, and it can't infect cells. So this is actually blocking binding of the virus to the cell surface. Again, you can get resistance rapidly, but this is used in combination therapy. Last question. Which of the following HIV antivirals inhibits the earliest stage of infection? Nucleoside inhibitors, NNRTIs, CCR5 inhibitors, integrase inhibitors, or fusion inhibitors. So the answer is CCR5 inhibitors. That blocks binding, which is earlier than any of these other inhibitors. We now use combination therapy to treat HIV-1 infections, and this is called highly active antiretroviral therapy, and you can live an entire life being HIV infected with these drugs because they keep virus at very low levels. And we use a mixture of 3 different inhibitors that target different mechanisms. Again, this doesn't cure your infection. You still have viral DNA in you. If you stop taking your triple therapy, virus will come back within a week. So you can't stop it for your entire life. Now, why does this triple therapy work? Here are the numbers. If you need 1 mutation for drug resistance, the mutation rate of HIV is 1 in every 10 to the 4th base is polymerized, so each base is substituted in every 10,000 viruses. Each person makes 10 to the 10th new viruses per day, so 10 to the 10th divided by 10 to the 4th, you make 1 million viruses every day with resistance to 1 drug. That's why you get drug resistance very quickly with monotherapy. How about double therapy? Developing resistance requires the product of the 2 mutation frequencies, 10 to the 8th. And so you're making 100 viruses resistant to 2 drugs per day. So it's a little better, but eventually you're going to get resistance. But 3 drugs, you need 10 to the 12th viruses, which is more than you make. And remember that when you start taking them, they inhibit reproduction. So these numbers are actually lower in the treated person. So triple therapy works. However, there are viruses out there that you can pick up that are triple drug resistant. And sometimes people get infected with them, and hopefully you can find another combination that will treat them, but we only have so many of them. In fact, here are all the drugs that are available for treating AIDS, starting with AZT, which was licensed in 1987, 3.5 months to approval. So this was fast-tracked because patients demanded it. They said, we need you to approve these. And all these are pretty much fast-track approvals. And you can see there are nucleoside inhibitors, non-nucleoside inhibitors, protease inhibitors, fusion, entry. We didn't talk about all of these. Integrase inhibitors. And then you have your triple combinations. The first one was a tripla. And then there are others, Complera and Stribild, and there are others as well. And as I said, they will tailor your therapy depending on the mutations in your virus. So remember, these don't cure you. You still have integrated viruses. So until we figure out how to cure people, you still have to take these drugs the rest of your life, but that's better than dying, of course. And sometime, as we'll talk about later in our AIDS lecture, we may figure out how to get rid of those proviruses from people. And as I said earlier, some of these can be used to treat hepatitis B virus, some of the reverse transcriptase inhibitors. Treatment with antiretrovirals saves people's lives, and as the years have gone by, we are getting drugs to more and more people who need them. You know, initially they were only available in US and Europe, but now we are hitting the African region, which has the burden of HIV globally, and you can see we're increasing the amount of antiretrovirals that gets there. This is an old graph, but the numbers keep increasing, and we save people's lives. Here we have 4 million adult deaths averted by treatment with ARTs and 800,000 child infections averted as well. And remember, these are not just adults, but you can treat pregnant mothers with these drugs and prevent the babies from being infected. We now do pre-exposure prophylaxis for HIV. It's the only virus where we treat healthy people with drugs, and that's PrEP as opposed to post-exposure prophylaxis, which I've been telling you about, of course. And this is double therapy, 2 drugs, and typically tenofovir and emtricitabine for people at high risk for HIV infection. So many countries of the world, you don't have a choice about having sex or not, or having protected sex, and this is for some of those people. And then we have learned that in other parts of the world, people take risks anyway, so we might as well give them a drug that will protect them. And this reduces the risk of sexual transmission by over 90%. It's actually better than a condom, but the 2 together are really good. And it reduces the risk of transmission by intravenous drug use by over 70% as well. There, IVDU is tough because you're putting a lot of virus in directly into the blood, so it's harder to block it. But again, taking these on a regular basis has been shown to be extremely effective. And of course, you don't have resistance because you are dealing with a very small inoculum. Typically, we think only 1 virus particle actually gets in the host and replicates. So these drugs can work better instead of having to deal with billions and billions of virus particles produced. But as I said in the first day, there are 10 to the 16th HIV genomes on the planet. In that number is resistance to all the drugs we have. You could sequence all of these and find resistance to every one of those 50 or so drugs already out there. And if we made more, if we made one a day for the next 100 years, we'd still have resistance. That's how big that number is. So next time we'll start to talk about how viruses evolve.